I'm going to ask you to stay with me for another hour or so. I know it's long, but you get 2 rewards. One is that lab this week is only 1 hour, and I've already emailed you about the hour that you should come to lab and the hour you don't have to be in lab. So that's one reward or whatever compensation for staying with me for another hour here. And the other compensation that I'm announcing right now, and I will send out an email reminder, is that on Monday, this coming Monday, I am teaching my neurophysiology lecture in cell physiology for the first hour of this afternoon slot. So Monday, we will start A&P at 1:45, not at 12:45. So you will have an extra hour not being in this room on Monday, okay? So stay with me for the next hour or so. And remember that you get that hour back, like daylight savings, you get that hour back on Monday. I'll send an email to remind you that on Monday we start at, let's just say 1:45, not 1:40, because I'll be teaching an hour or so in self-phys. It's my last time I will be teaching that neurophys lecture in self-phys. I'm full of nostalgia, but it'll be weird, but I'm going to do it anyway. Okay, you take a bite of food and chew. What happens? The top 3 happen, but protein is not enzymatically digested in the mouth. Fat and carbohydrate are. You swallow a bolus of food. What happens? Complex response, reflex, moving the food into the oropharynx and into the, out of the mouth and down into the esophagus. The epiglottis closes. Food is propelled in the esophagus by peristalsis, not by gravity. Not by gravity. Questions about these clickers? The stomach secretes acid. What happens to plasma pH? I already did this. It increases. Okay, let's do the Davenport diagram here. Too distracting for this. All right. I'll start. What next? Just shout it out. Anything.
Yeah.
Sarah, I see you're dying to shout out something.
We can put plasma pH on the x-axis.
Okay, plasma pH. Down here. What else? Somebody else? Come on, don't all shout at once.
Plasma bicarbonate on the line.
Okay, plasma concentration of bicarbonate. Okay, we're not there yet, but we're getting there. Did I see a hand in the back there? Yes, please.
You can put like the normal resting point or.
Okay. Okay, now, any more?
At like the 7.4 pH.
Okay. 7.4 is the normal resting pH, and what's the normal plasma bicarbonate?
26.
26. What are the units there? We know I care about units. Millimolar, or because it's bicarbonate, we could call it milliequivalents per liter, but we'll just call it millimolar. Okay, so Very good. Need more dots after that. Here's our starting point. Okay, what next? Sophie.
You can draw the curve for like PaCO2 at 40.
Okay, curve. Curves up this way, not the other way. PaCO2 40, what are the units?
Milliliters per mercury.
Milliliters of mercury. All right, there, that's a Davenport diagram, a well-labeled Davenport diagram for the normal. What is the effect of digestion? What's the effect of gastric acid secretion during the gastric phase on the Davenport diagram?
Metabolic alkalosis.
Okay, metabolic alkalosis. What does that look like on this diagram?
Just
You're
up.
correct. Which way do I go?
Up.
Up this curve?
Yeah.
Up this curve. Metabolic. This one is good. Metabolic alkalosis. pH is getting alkaline because the stomach is getting acidified. Okay, good. There's the metabolic alkalosis. Metabolic alkalosis. Okay, what's the renal response to metabolic alkalosis?
The arrow going down.
The arrow reversing. So the arrow going down would be the renal response to metabolic alkalosis. What is the kidney doing?
Secreting bicarbonate and retaining pH losses.
Is bicarbonate excretion through the kidney increasing or decreasing? The bicarbonate is decreasing. So the excretion of bicarbonate is increasing. So this is the renal compensation. It would increase the excretion of bicarbonate. Okay, let's just say it's only partial. What is the respiratory response to metabolic alkalosis? Hypoventilation. What does that look like on this Davenport diagram? Hypoventilation. So that's the respiratory compensation. And it's moving to a curve with a PaCO2 higher or lower? Higher. And those things both happen during the gastric phase of digestion. You may not be aware of your lungs changing respiration, but they do. You breathe more slowly. Your tidal volume is negatively affected by the contents of your stomach. You can't inhale as deeply as you can override it, but a normal ventilation, your tidal volume under normal ventilation would be decreased if the stomach is full of contents. This is what happens when you digest a big meal. So both of these arrows are happening, and so what's really happening is probably some combination. It probably goes this way, and then it goes this way, and then it goes back to the starting point. Well, back to the starting point. Right? There's some combination of respiratory compensation and renal compensation that brings us back by some circuitous route back to the starting point. Okay, good. Be prepared to do something like that on the next exam, not on the quiz. So now, let's go to the last. Let's go to the last PowerPoint. And start to talk about the pancreas and the liver. Okay? So we're moving on. We're going to learn about the exocrine pancreas, not the endocrine, not insulin or glucagon. We're going to learn about the exocrine secretions of the pancreas. New word, zymogen is the generic word for ogens. So pepsinogen is a zymogen. And we're going to introduce some other ogens, which are not ogens, they're zymogens. Zyme from enzyme, ogen for precursor. Enzyme precursors. And bicarbonate for neutralization of gastric acid. We're going to learn about their control by the hormones, CCK and secretin. We've introduced those before because those inhibit gastric acid secretion, but they are going to stimulate pancreatic secretions. We're going to talk about proteases, and then we're going to start to do liver anatomy. And if we get through all that, we're good. With me still? A cartoon of the pancreas. This is the loop, the first loop, the curve of the duodenum. So here's the duodenum. The pyloric sphincter is here, the stomach is here, but not shown. So the stomach, pylorus and pyloric sphincter, entry into the duodenum. The pancreas is tucked into this curve of the duodenum. There's a head and a tail. We're not going to worry about that. The pancreas on our specimen up in the cadaver lab doesn't look like this. It's just been shrunk. The pancreas, the exocrine pancreas, like the exocrine salivary gland, has acinar cells, and duct cells. Your book calls them exocrine cells. It's all exocrine, so I don't understand why they call them exocrine cells. I'm going to accept exocrine cells because it's in your book, but they're called acinar cells. The acinar cells are the cells that secrete the enzymes, the zymogens. They don't secrete enzymes, so your book has some mistakes here. The acinar cells secrete zymogens, and the duct cells secrete bicarbonate. This duct, so here are the cells out here in the pancreas, and so this microscopic duct, remember this is an epithelial cell, so this is cellular in dimension. And these tiny little ducts are what connect in these little nubs here to the pancreatic duct in the pancreas. So the pancreatic duct has a name because it's big, and it's getting filled with pancreatic juice coming bicarbonate and enzymes, bicarbonate and zymogens being secreted by all these little glands into the pancreatic duct. The pancreatic duct ordinarily has two points of exit into the duodenum. One of them, they both have names. I'm going to ask you to know the name of this one. Well, I'm going to ask you to know the sphincter that controls the opening and closing of this duct. It's called sphincter of Oddi. So the sphincter of Oddi opens to allow pancreatic secretions, and it turns out to allow bile from the liver and the gallbladder to be emptied into the duodenum. So that's the plumbing that I want you to know. The pancreatic duct, the sphincter of Oddi, the bile duct from the liver and gallbladder. The enzymes that are secreted by these, enzymes and zymogens that are secreted by the acinar cells of the pancreas include amylase, that's an enzyme. Lipase, that's an enzyme. Carboxypeptidase is an enzyme. Chymotrypsinogen is a zymogen, as the name would suggest. It's the zymogen, it's the ogen of an enzyme called chymotrypsin. And trypsinogen, the precursor zymogen for trypsin. Amylase, you know, is important for the digestion, the breakdown of carbohydrate, starches into mono and polysaccharides. Lipase, the breakdown of fats, which we'll talk about on Monday. Carboxypeptidase, protein. Chymotrypsin, another protease. This is digestion of protein. And trypsin is an enzyme that breaks down protein, peptide bonds. It's another protease. These are the enzymes that I do want you to know the names of, that are secreted by the exocrine pancreas. Questions about what to know and what not to know on this cartoon? Here is an EM, an electron micrograph of a pancreatic acinar cells. So it says acinar cell, a lumen chock full of secretory vesicles, full of the things that are secreted. The zymogens, the enzymes, just a dense, that's why they're black on an electron micrograph. They're so full of enzymes that they are electron dense. All the rest of this stuff, you know what that is. That's endoplasmic reticulum for the making of these secretory vesicles. These are all peptides. Remember, they're enzymes, so they're proteins. So they are all using the co-translational translation. Remember that story from Self is? Co-translational translation of mRNA protein into ER, which then gets processed by the Golgi into secretory vesicles. Let's talk about the control of release of these enzymes. The control is a hormone from the duodenum called CCK. I will not ask you to know this, but the name CCK comes from cholecysto. I'm not sure if that's an A or no. Kinin. Cholecystokinin. C. Okay, that's where the name comes The name comes from the fact that this hormone causes the gallbladder, in addition to triggering the secretion of these hormones, enzymes, enzymogens, this hormone triggers the gallbladder to squeeze and squirt bile. And that's where the name comes from. Because the cholecystokinin, kinin means to move, cysto is referring to the gallbladder. So the name, if you know Latin, this name makes some sense. So the name comes from the fact that CCK, in addition to doing this, triggers the gallbladder to squeeze and squirt bile. CCK triggers the gallbladder to squeeze and squirt bile into the intestines, and it causes the pancreas to secrete enzymes and zymogens. That's what CCK does. And CCK does that, well, there's the flow chart. The small intestine detects the presence of chyme. It detects the products of partial digestion. The duodenum detects chyme entering it. The endocrine cells of the duodenum secrete CCK. CCK goes into the bloodstream and responds and binds to receptors on the gallbladder, which triggers squeezing and contraction, and on the pancreas, which tells the cells to secrete their contents. Yes?
Is the contraction of the gallbladder a GQ story or
Yes, it is.
cross recycling?
Yep. CCK receptors are GQ linked, just like gastrin receptors. In fact, you may have noticed that the gastrin receptor and the CCK receptor are almost identical. So, fatty acids and chyme, amino acids and fatty acids in the small intestine trigger intestinal endocrine cells to release CCK. CCK goes everywhere, it's a hormone. It binds to CCK receptors in the pancreas and triggers secretion of enzymes and zymogens. And, not shown here, it binds to the same receptor on gallbladder smooth muscle, triggering contraction, GQ story, contraction, squirting of bile. Bile and enzymes are necessary for the digestion of fat and protein. So here is that story. Here's CCK binding to GQ receptors, triggering the secretion of enzymes. So that's the part of this story I want you to know. CCK, GQ, you're off and running. Secretion of protein kinase C, secretion of enzymes and zymogens. In the smooth muscle, this is not smooth muscle, this is a acinar cell of the pancreas, but in smooth muscle of the gallbladder, calcium, where's calcium? Calcium binds to calmodulin, myosin light chain kinase, we're off and running with the smooth muscle contraction. The other major hormone that we're talking about now is secretin. Secretin is coupled to GS, which also triggers release of enzymes and zymogens through the PKA GS story. Really, what happens here is that the main stimulus is CCK, and a auxiliary supporting stimulus is secretin. So CCK stimulates secretion of enzymes and zymogens, and secretin augments that. Okay? Now we're going to turn our attention to the secretion of bicarbonate. What cells secrete bicarbonate? Duct cells. Duct cells of the pancreas secrete bicarbonate. How is that controlled? That is primarily controlled by secretin. So acid in the stomach, or acid from the stomach, getting into the intestines, gastric acid, pH detected, and hormone endocrine cells in the duodenum secrete secretin, bind to secretin receptors on the duct cells and trigger the exocytosis of the transporters necessary to move bicarbonate. We'll see that in a moment. So, the primary stimulus for duct cell release of, or secretion of bicarbonate is secretin, but CCK augments it. So CCK is primary for the secretion of zymogens and enzymes, and secretin augments it. Secretin is primary for the secretion of bicarbonate from the duct cells, and CCK augments it. They both have a role, but one is primary and one is secondary. You could predict how these duct cells secrete bicarbonate. You could probably already write it out, but I'm not going to make you do that. I'm going to just show it to you. So here's another one of those complicated slides. I think I gave this one to you. I gave it to you big, but you really don't need to know most of this. Here is the chloride bicarbonate exchanger on the apical membrane, because this is apical because it's facing the duct. Here is the chloride bicarbonate exchanger. Here is the sodium bicarbonate cotransporter. You know that the bicarbonate concentration out here is around 26 millimole. So sodium bicarbonate cotransport and carbon dioxide conversion into bicarbonate and bicarbonate chloride exchange pump bicarbonate into the duct movement. Same players as in the kidney, just running slightly in reverse. Sodium bicarbonate in the basolateral membrane, but in the kidney, we were running this the other way. Chloride bicarbonate exchange used to be, in the kidney was in the basolateral membrane. Now here in these cells, we're putting it in the apical membrane, and we're using, well, we're allowing the bicarbonate to leave into the duct. What does secretin do? Remember there are vesicles here with this protein sort of resident here, and secretin increases the exocytosis of those vesicles with this transporter in it, so that it increases the transport of bicarbonate. The rest of this we don't have to worry about. I mean, here's the sodium potassium ATPase, here's the proton pump, here's the sodium proton exchanger. These are all your good friends from the kidney, but we're just not going to focus on them. You can imagine what is responsible for water getting through this membrane. Say it. Aquaporin. You can imagine that water has to get through this membrane too in order for this juice to have some water in it. and that's aquaporin as well. Constitutive activity of aquaporin, water for the production of bicarbonate and water for the production of fluid, is through aquaporin. This is the main story, right here. That's what I want you to know about how pancreatic duct cells secrete bicarbonate. Prevention?
How is the water permeability different now from the kidneys? Because in the apical membrane of what cell are we talking?
Oh, so
I just want to know the water permeability. Do we have a bunch of aquaporin?
we have a lot of aquaporin constitutively active on both membranes here. So this is not vasopressin dependent. I could walk through more of the story, but that's not more than we need. So let's look through this slide again. And let's focus on CCK and secretin now. CCK is a peptide hormone, and so is secretin. Both of them are secreted by endocrine cells in the duodenum. The stimulus for CCK secretion is the products of protein and fat digestion. And the stimulus for secretion of secretin is gastric acid. All of those things are getting from the stomach into the lumen of the duodenum, and that's triggering the release of CCK and secretin. Both of them inhibit, let me use red here, because this is an inhibition. Both of them inhibit the production of gastric acid. This is negative feedback. Once the chyme is getting into the intestine, these hormones are telling the stomach, cool it, stop working so hard. CCK stimulates by enzyme secretion. Secretin, now let me do this in Greek. CCK stimulates enzyme secretion. Secretin stimulates bicarbonate secretion. Enzyme secretion by the acinar cells, bicarbonate secretion by the duct cells. CCK potentiates, augments secretin's action, and secretin potentiates CCK's action. CCK is the stimulus. Can you even see this down here? This is the stimulus for gallbladder contraction. So those are the things that I want you to know about CCK and secretin. The rest of the things on the slide, like motility and growth, and insulin and exocrine growth, just leave alone. We'll get to the liver on Monday. Questions about what to know and what not to know? Yes?
For clarity, the duct cell, or the secretin comes from the duct cell?
No, secretin comes from endocrine cells in the duodenum. Secretin's site of production is the small intestine, and And since it's a hormone, it has to be coming from endocrine cells in the small intestine.
Thank you.
This is where it's coming from, and this is where it is acting on. And this is where it is acting on. It is inhibiting the stomach secretion of gastric acid, and it is primarily acting on the duct cells to make bicarbonate. CCK is coming from the small intestine, it inhibits the stomach, and it is stimulating the acinar cells to secrete enzymes and zymogens. Okay? Okay, one more thing, at least one more thing, and then maybe we'll be done. We'll see. Let's talk about those enzymes and their zymogens. So this is a cartoon of the pancreatic duct emptying into the duodenum. What's the name of this sphincter that controls pancreatic juice flowing into the lumen of the intestine where chyme is coming from the stomach? What's the name of this sphincter? Oddi. Sphincter of Oddi, it's right there. Okay? So there is a meal, there is chyme in the intestine that has been squirted in here from the stomach. Let's just say it's a meal that has some fat in it, it's got some protein in it, but it certainly has some acid in it, right? Because gastric acid. The amino acids from the protein stimulated CCK. The amino acids from the meal and the fatty acids from the meal stimulated CCK production. And that told the pancreas secrete enzymes, enzymogens. The gastric acid from the intestinal endocrine cells told the duct cells of the pancreas to make bicarbonate. Bicarbonate, enzymogens, and enzymes are being secreted along with water. Sphincter of Oddi relaxes, juice flows into the lumen of the intestine. The juice contains a zymogen. I forgot to mention this. So carboxypeptidase is an enzyme, and it has a zymogen that's called procarboxypeptidase. Chymotrypsin is an enzyme, and it has a zymogen called chymotrypsinogen. Trypsin is an enzyme, and it has a precursor, a zymogen called trypsinogen. Okay, so we have zymogens, trypsinogen, chymotrypsinogen, and procarboxypeptidase in the pancreatic juice. Resident in the epithelial cells of the small intestine, of the duodenum, is an enzyme that kicks this all off, called enterokinase. Now, this is a really bad name. Despite its name as kinase, it's a protease. This enzyme is a protease. It cuts trypsinogen to trypsin. So, just like in the stomach, where acid cut pepsinogen into pepsin, which then did a positive feedback loop, pepsin was making more pepsin by hydrolyzing more pepsinogen. There was that positive feedback. Remember that just a couple hours ago? Enterokinase triggers this by making trypsin, and trypsin can hydrolyze trypsinogen. It's not shown here, but trypsin can hydrolyze chymotrypsinogen, and trypsin can hydrolyze procarboxypeptidase to make the final enzymes. So enterokinase starts it, trypsin, positive feedback. These enzymes they're proteases, they're all proteases, and so they proteolyze the protein in the meal, and they proteolyze the ogens, the zymogens, to make more enzyme. Positive feedback. We generate a lot of enzymes for the hydrolysis of peptides, breaking peptide bonds, turning protein into amino acids. So we have turned carbohydrate into monosaccharides by amylase. Well, we haven't gotten to monosaccharides yet. We've cut starch into oligosaccharides with amylase. We have cut protein into amino acids with these enzymes in the small intestine. So, what is not shown here? Amylase is one of those pancreatic enzymes. So it should be, we should also have amylase here. But amylase is not a ogen, it's ready to go. It's ready to start hydrolyzing starch. What else is in here? Lipase. Lipase is ready to go, start hydrolyzing lipid into lipid breakdown products. So here is the cartoon. We have the pancreas doing the stories that I've just been telling you. The duodenum, both the source of CCK and secretin that tells the pancreas to do its thing, and the recipient of those enzymes and bicarbonate. The bicarbonate to neutralize the gastric acid, and the enzymes to do the hydrolysis of carbohydrate by amylase, lipid by lipase, and protein by carboxypeptidase, trypsin, and chymotrypsin. That's all happening now, and the chyme is going to continue to move down the intestines, continuing to be digested, breakdown products being reabsorbed, and the absorption we'll talk about on Monday. Let's finish with a little anatomy, just a little more anatomy. So pancreas, duodenum, stomach, not shown. Gallbladder. I want you to know the names of some ducts. The pancreatic duct. The bile duct, which is sometimes called the common bile duct, but I will accept bile duct. Pancreas, bile duct. The bile duct, comes from the junction of the duct from the gallbladder and from the liver, of the cystic duct from the gallbladder, and the hepatic duct, the common hepatic duct from the liver. So from the liver, we're going to see a left and a right hepatic duct. So the names sort of help you. Left and right is just the side. Left hepatic duct, hepatic just means liver. Left hepatic duct, right hepatic duct, join to form the common hepatic duct. The common hepatic duct and the cystic duct from the gallbladder join to form the bile duct. And the bile duct empties into the duodenum. Between meals, like right now for most of you, the sphincter of Oddi is closed. Pancreatic juice is not flowing. bile is being synthesized by the liver, but it's backing up, this is closed, the bile is backing up into the gallbladder and being stored between meals. Closed, production, storage. When you eat a meal, secretin and CCK primarily, remember, stuff from the meal getting into the duodenum, endocrine cells, CCK opens the sphincter and squeezes the gallbladder, juice squirting, bile squirt during the digestion of a meal. Okay? So in the PowerPoint, there's some of this game which you can use to test, to help you. I'm going to leave this up. Did I give you this slide? Yeah, let's do this one so that you have the answers. And things that I want you to know anatomy-wise from something like this. So this is the liver. What lobe is this? The right lobe. And this is the left lobe. If we were looking at the back, we would also see some more lobes like the caudate lobe. But this is the anterior perspective. Left lobe, right lobe. What are these? Left and right hepatic ducts. What is this? Common hepatic duct. Right hepatic duct, left hepatic duct, common hepatic duct. Right lobe, left lobe. So far so good? Pretty easy so far. What's this? Cystic duct. What's this? Gallbladder. Gallbladder, cystic duct, left and right hepatic duct, common hepatic duct. What's this? Bile duct or common bile duct. I'll accept it either way. Bile duct or common bile duct. What is this? Sphincter of Oddi. In the previous slide, there's another name. There's the ampulla of Vater. It's like, okay, how many names? Whatever, okay? I don't care if you know this name. I do want you to know the name of the sphincter. Sphincter of Oddi. It's got another name. So, I mean, how many names do we need for this tiny little thing? Sphincter of Oddi. Sphincter of Oddi, right here. What's the name of this tube? Pancreatic duct. Pancreatic duct. Pancreatic duct and the bile duct both join at the ampulla, but whatever, sphincter of Oddi opens and closes, and when it opens, bile and pancreatic juice are delivered to the duodenum. What's the name of this? I just said it. Duodenum. The pancreas has a head and a tail, whatever. What is the name of the sphincter that opens and closes that allows chyme to move from the stomach into the duodenum? pyloric sphincter. Okay? I think, oh, one more bit of anatomy. What is this thing? It's a ligament. It's called the falciform ligament. You saw this on the cadaver, holding the liver in place. That's the liver anatomy that I want you to know. I think, what else? Gallbladder, we don't care, you know the gallbladder. We'll get to the jejunum, that's the next segment of the small intestine, we'll get to that on Monday. Cystic duct we did, gallbladder we did. That's the sphincter of Oddi. Bile duct we did, forget that. Pancreas, whatever, it's got a head and a tail and a body, whatever, the pancreas. And I'm sorry, when I do this whatever thing, it just shows that I am not a dyed-in-the-wool, hardcore anatomist. There are things that I do want you to know, and there are things that only if you become a surgeon working on gastrointestinal surgery do I think are really important. So for those of you who are, maybe your parents are anatomists and they're tearing their hair out because you're not learning it right, but I'm sorry. Pancreatic duct, we talked about that. Okay, so these two slides, I think I've given you the answer to the things that I want you to know on a slide like this. It's anatomy. We've covered it. What does that mean? It could be on the quiz. The next thing we're going to do is the functions of the liver, and I'm just going to hide the slide where that is not on the quiz, because we're not going to cover it. Okay, we'll do that on Monday. It'll be on the PowerPoint, that's up on Moodle, but we're not going to do it. Questions before I send you on your way? I'll see you on Thursday. Remember to come at the time indicated on the PowerPoint. No, in the email. There's an email. It's not loaded up onto Moodle. If you have a question, just let me know, but it's in your email what time to come. Don't be late. We'll do 1 hour with the cadaver. In the cadaver, we're going to look at the gastrointestinal, we'll look at the digestive system. I will give you some tips about how to prepare for the practical, which is coming up next week. Practical quiz. So we've got a quiz this weekend. We've got a practical quiz next week. One more exam. We're done. Question?
Practical in lab time, or is it useless?
It's going to be in the lab time. So I will talk about it when we're in lab tomorrow or Friday. Sophie?
Does the quiz have physiology then? Like
The quiz-
Can you do or no?
The quiz has names of both anatomical and physiological things, but it is not going to ask you for mechanisms. So it might ask you the name. What is the enzyme that is primarily responsible for the secretion of bicarbonate? And you would have to say, okay, that's secretion. What is the name of the enzyme that's primarily responsible for the squeezing of the gallbladder? CCK. How does CCK trigger the secretion of enzymes from the pancreatic duct? Not. Okay, so it's got names of things that are anatomical and physiological. Is that clear? Clear enough? Okay.